Today, we're going to flip a switch on how we think about life's relationship with light. We're going way beyond photosynthesis to explore a different and much more ancient light switch, the incredible microbial rhodopsin.
Okay, so let me ask you something. When you think of organisms using sunlight for energy, what's the first thing that pops into your head? It's photosynthesis, right? Green plants, algae, chlorophyll, the whole deal. But what if I told you that's not the only game in town? What if there's another, even more widespread way that life captures light?
We'll say hello to microbial rhodopsin. And it's not just one thing, it's a huge family of proteins that you could find all over the tree of life. The best way to think of them is as tiny light-activated biological machines. The second a photon of light hits them, bam, they spring into action, turning that light into either a little jolt of energy or a crucial piece of information for the cell. And what's so fascinating is just how elegant their design is. I mean, even though they have all these different jobs, they're all built on the exact same fundamental blueprint. It's this beautiful scaffold of 7 protein helices that weave back and forth through the cell membrane. And right there, nestled in the middle, is a special molecule called retinol. That's the light-catching antenna. And this simple shared structure, well, it's the key to everything they do. So here's what we've got on tap. First, we'll see just how common this other light switch really is. Then, we'll meet the incredibly diverse rhodopsin family. After that, we'll take a peek under the hood at how these pumps, channels, and sensors actually work. And we'll wrap it all up by looking at how this ancient microbial machinery is literally changing our world today.
All right, let's jump right into our first section, Life's Other Light Switch. We're about to uncover a totally hidden world of phototrophy, which is just the fancy word for using light for energy.
You see, these proteins aren't some rare little curiosity. No way, they are everywhere. Scientists have found them in all 3 domains of life, in ancient microbes called archaea, all throughout the bacterial kingdom, and even in eukaryotes like fungi and algae. They're a truly fundamental part of life's toolkit for dealing with light.
And this is where it gets absolutely wild. Get this. In the world's oceans, scientists estimate that microbial rhodopsins are about 3 times more abundant than the machinery used for photosynthesis. Let that sink in. 3 times. This is not some little backup system. It's a major, major player in how our planet captures energy from the sun.
Okay, so for our second section, let's meet the family. Remember, they all share that same basic toolkit, those 7 helices and the retinol molecule, but evolution has tinkered with them to do a stunning variety of different jobs. We can basically sort them into 3 main groups. First up, you've got the ion pumps. When light hits them, they physically push ions, like protons, across the cell membrane. This creates an energy gradient the cell can use for power, kind of like a microscopic rechargeable battery. Then you have the photosensors. These guys don't move ions, they act more like detectors. They absorb light, change their shape, and send a signal that tells the microbe to, say, swim towards the light or away from it. And finally, you've got the channels. Just think of these as little light-activated gates. A flash of light makes them pop open, letting ions rush across the membrane.
So what's the engine driving all of these different functions? Well, it all comes down to one single incredibly fast event. When light smacks into that retinol molecule we saw earlier, it causes it to instantly flip its shape. This tiny light-speed molecular twitch is the trigger. It's the starter pistol that kicks off the protein's entire mechanical cycle, whether it's pumping, signaling, or opening a gate. Which brings us to section 3. Let's take a closer look at the mechanics of how these different types actually get the job done. How do they work?
Okay, let's start with the classic proton pump. When light hits it, that retinal flip we just talked about triggers a whole cascade of shape changes in the protein. This motion effectively grabs a proton from inside the cell and shoves it outside. By doing this over and over, it builds up a powerful energy gradient that the cell can tap into. Now, other rhodopsins do pretty much the exact opposite. They use the same light-powered mechanism, but instead of pumping positive protons out, they pump negative chloride ions into the cell. It's just a beautiful example of how evolution has tweaked the same basic machine to do totally different things. And finally, we have the sensors. These are the information gatherers of the family. The retinol still flips, the protein still changes shape, but it doesn't move any ions. Instead, that shape change is recognized by a partner protein, which then relays a message deeper into the cell. So it's not about creating energy, it's about collecting information. It's basically a light-activated alarm.
And this all leads us to our final section, which is, well, it's a big one. We're going to see how these amazing tools went from helping microbes survive to powering human technology. I mean, the potential here is just staggering. Because these proteins are basically light-switchable nanomachines, researchers are looking at them for all sorts of wild applications. We're talking new forms of energy conversion, new ways to build computer memory and chips, and even a completely revolutionary field called optogenetics. This quote from one of the pioneering research papers really says it all. The ability to depolarize the cell membrane just means you can activate a cell, like a neuron in your brain. And the incredible part is, you can do it with insane precision, simply by illumination. You can literally turn a cell on with a flash of light. And that is exactly what optogenetics is. Scientists can now insert the gene for one of these rhodopsins into specific brain cells, giving them precise control over those cells. You want to know what a certain group of neurons does? Just shine a light on them and watch what happens. This technique has absolutely, completely revolutionized neuroscience. So we'll end with this thought. We've discovered literally thousands of different microbial rhodopsins, and each one is a slightly different tool with its own unique properties. We've already turned one of them into a way to control the brain. So the big question is, with this massive natural library of tiny, light-powered machines at our fingertips, what amazing new technologies are we going to build next? Thanks for joining this explainer.